The work that I talk about today, it's targeting the vasculature, but not in the classical point of view, as it has been done so far, but it is to instruct the immune system to recognize specifically the vasculature of the tumor and kill the already existing vasculature in the tumor. And so it's a different approach of the classical anti-angiogenetic therapy. Indeed, because the anti-angiogenetic therapy blocks the formation of new vessels. Vice versa, this approach has the characteristic to attack the vasculature of the tumor that is already there. We are demonstrating that indeed this vaccine is capable to induce a T-cell mediated immune response, which indeed target the vasculature of the tumor, destroying the vasculature, inducing ischemia and inducing necrosis, apoptosis of the tumor cells. On top of that, really interesting is this phenomena of cross-priming or epitope spreading that we are observing that means that killing the cell indirectly, because we don't target the cells directly with the vaccine, but killing the blood vessel, blocking the furniture of the food and the oxygen, the tumor cell die, and then after that phenomena, the tumor cells are also engulfed by the antigen-presenting cell, by the immune system cells, and those antigen-presenting cells then, in return, expose the tumor-associated antigen. So those proteins that then are capable to elicit a secondary immune response against the tumor itself. So we have a two-pronged approach using only one vaccine. At the moment we tested this with several tumors. This is also another exciting feature of this approach. Indeed, the TEM1, it's expressed, that is the target molecule, that is Tumour Endothelial Marker 1. We used 4 different models of cancer, including breast, including Lewis-Lung carcinoma, including cervical cancer, and including a colorectal cancer model. And in all of them we obtained the same results. We were capable to block the formation to destroy the already present vasculature, block the formation of new vasculature, and on top of that induce the secondary immune response against the tumor itself. And in all of those different models we obtain similar results. And this is a really nice thing, because this will give us the chance to target several cancers with one single approach. Yeah, we did actually. There is all the work done because angiogenesis is a physiological process which is important for wound healing, pregnancy, and therefore, of course, we went to look if our therapy would have impacted the pregnancy or wound healing, and indeed in animals we didn't find any impact. There is another piece of information that I want to give it to you. There is a knockout animal, so it doesn't express TEM1, and it doesn't have impact on wound healing or pregnancy also, confirming our data and telling us that indeed targeting TEM1, it seems to have no side effects on adult physiology, angiogenetic physiology. The take-home message is that we are moving forward in the therapy of the cancer. There are several approaches that have been developed, and the idea to target the vasculature through the immune system, it's also extremely exciting, because it's not like the other compound that have been developed up to now, which is specific for the vasculature, but it's active only in the moment you administrate the drugs. This is going to be an immune response, therefore memory, therefore long-term protection. So the take-home message is that with immunotherapy we can obtain not only a reduction of the tumor or cure in one instance, but we can get protected for the rest of our life once we induce this immune response against the specific molecule that we are looking for in the cancer.